Yeah. So okay. So first understand what exactly this package, what we can do with this package. So this is a collection of Python tools, or we can say Python functions, different functions, which is required for computational biology and bioinformatics. Means whatever the functionalities we need to work with biological data, like data in the sense you can handle DNA sequence, RNA sequence, protein information, genes expression data, these all things. So how to manipulate or how to do analysis, right? Okay, so for that biopython package is very important. By using this you can create a sequence, which we already learned in how to create a sequence, or how to create a string data, like text data, character data. So the similarly you can create a sequence with help of biopython, that is a string representation, the sequence as an argument, and all other functions, the simple, that is like a transcription, translation. Then there is a reverse complement, reverse sequence, if you want to count individual nucleotide base, that even GC content, or what we learn in that, finding subsequence. Okay, so find method also you can use to find the subsequence, like a motif or primer, if you want to search in that sequence. And also concatenation of the sequence, means connection of more than two sequences put together. So string representation, these all things we can do as a string manipulation. So first thing, as I told, most of the packages are already installed, right? So here, which are installed, those are widely used for data science application or machine learning, or mostly for statistical analysis. But this package is specifically for bioinformatic application. So this is not already installed or pre-installed in this cloud. So we need to install by command pip install. You can write biopython. This is the name of package. So you can try this command. So what all means we can do with this? So there are many modules. Okay, let me pip install biopython. Just wait. Yeah, I think there is a comment, so it's not working. But here you can see it's collecting biopython. Previously, when we try pip command, that time we are getting message. The command is fulfilled. Okay, the requirement is already fulfilled. But here it is showing message collecting biopython. Means it is downloading that package inside this environment, and then we'll get a message successfully installed biopython. Okay, this step is important whenever we are working with this package in this environment. After installation, we are ready with that biopython. So biopython, this library is basically for, as I told, computational biology or bioinformatics. And there are many modules, many classes which comes along with this package. Means what? Once we have imported, we have installed this, we can import many modules by using bio dot, when we want to work with sequence. So sequence means any kind of sequence, so that time you can call sec module like this, bio dot sec. B should be capital. Okay, so bio.sec, with this we can work with sec function. By using sec, you can represent DNA, RNA, protein sequences. And for that, there is a method for manipulation analysis. Also, we can work with mutable sequence, mutable sec. Basically, this is similar to sec, but it allows for the modification of the sequence after creation of the sequence. Here you can see mutable sequence. Okay, why we are getting these all things? Because we already imported this biopython. That's why from typing import mutable sequence and all these things, auto downloading. Yeah, we are not working with this. So let me comment this. Let's first understand what all modules are there. So second is there biosec record, bio dot, here sec record is the class. So with help of biosec record, we can, this is basically nothing but metadata. Are you aware about this term, metadata, or annotation, or uh, data annotation features? So any detailed information about that database, about that sequence, is nothing but the metadata, like your annotation, your features, database references. So these all useful information which is handled sequence data in that context, so that is called as a metadata. If you want to do some alignment, okay, so alignment is again local global alignment or multiple sequence alignment. So this you can do with bio.align. If anybody is interested in protein data, then they can work with pdb dot, sorry, bio.pdb. So pdb means what? You already learned this in first module, right? This database. So if you want to extract any structural information of biomolecules or any crucial or important information, structural bioinformatics, if anybody wants to work, then they can use pdb files, like they can parse the pdb files to extract this all information. Even there is mmcif files, which is alternative format of pdf. So that also provide you structural information about the protein, that we can work with bio.pdb module. Then there is a Entrace. Entrace is what? You might use earlier this Entrace, right? So search engine, when you want to fetch some information, so you can use ePage class to retrieve records from database, Entrace database. Like if anybody wants to fetch from PubMed for literature, or if anybody interested in GenBank to search some sequence. So here you can use identifier, unique identifier or accession ID. Even you can search information. Searching is also possible through Entrace class. Then, okay, just tell me what is the use of Blast? Blast is also done, right? Online, offline, web based. So Blast, sequencing, no, Blast, yeah, for similarity search. Basically, Blast is used for similarity search. Fine. So if you want to do some search, sequence search, as as you told, so that time you can use Blast class. So again here you can use ncbi www for ncbi Blast web services, or ncbi standalone Blast also, like which is generated through Blast standalone Blast software, or local Blast analysis, we can say. So this is the things. Then also, if for phylogenetic study, like tree structure, or just want to represent tree, or the ancestral history, then they can work with phyllo class. So phyllo class is basically when you want to parse or write phylogenetic or xml files, which are used for storing phylogenetic tree, and that tree also having annotation information. So there is one more module, that is swissprot. Sysprot is again sequence data, and also annotation with the protein sequences. Also, if we want to do sequence input output, then there is a module seq.io, that is sequence read write in various format. So this also possible with biopython. Then if somebody wants to study pathways, then there is a keg. So KEG is basically pathways and gene information, which is very useful for pathways analysis, even for functional annotation, right? Also, there is a one class called as enzyme. Enzyme is basically like enzyme records, or any specific information regarding pathways, so that we can study through this enzyme class. In there are many other classes like restrictions, emboss. So this is belongs to biopython package, biopython library. So how to import this? Like first step is installation. We did this, pip install biopython. So now we are ready with this bio module, and through bio module these are different classes. So first we need to import that bio. So for that we can use, similarly yesterday we import different libraries like matplotlib, numpy, and all the thing. So two ways. Here we can use from, From biosec, so from biosec, let's work with sequence first. So I'm importing sec class. So this is basically sec is the class from bio.sec module, and these all things belongs to biopython library. So biopython is what? Biopython is a set of tools for biological analysis, biological sequence. So sequence in any form, DNA, RNA, protein sequence. Here you can create sequence, manipulate sequence, analyze sequence by using this sec class. Okay, this sec should be capital sec. This is all. You can also import this sec class. Okay, so let's do some analysis with this sec. First, let me show the difference. What is the difference between simple string for sequence string? Let's create one sequence string. Suppose here I'm creating one variable. This is variable. This is not class. You can see you can give any name, DNA sec, sec1, sec2, sec1, 2, 3. So this is just a variable which store a sequence. And here I'm creating sequence. This is a sequence or just a string. You can say this is a string, or let's check what exactly this sec. So here I'm calling sec, and how to check? Call type function. What is happening? Type of sec. So it is a string, simple string. There is one more function called as a dir. Dir means directories. Dir is basically what? Let's check directories for sequence. So directories means all attributes and a list of attributes or list of methods which are available with help of with that variable. We'll get all the detailed information. So let me execute directory for sequence. Here you can see underscore means these are all attributes. Even these are all different methods. Methods means functions. Like we can do capitalization, we can do case fold, center, count. So these all things we already did in third session. Third, that is string manipulation. How to count individual frequency. Then there are many things you can see here. List of methods available starts with, if you want to check any correct any word which starts with particular letter, so that you can use translate we check, upper we check. Okay, so it is not possible to execute each and every function. You can try for practice later. Here you'll get all detailed information how to use, what all parameters you can use. Okay, when you call this, you'll get this documentation. So is lower to check whether it is lowercase or uppercase. So these are all available with type simple string. Now we'll create one more sequence with help of this sec class. So for that what we need to call this sec function. So let me do one thing here. Let's create new variable called as biosequence or biosec, because we are using biopython here, and then call sec function. So this is a function. Inside function, let's pass this variable. This is a variable, okay? So I'm calling this variable inside this sequence, because we just want to, or you can directly use this sequence. It is also okay. Okay. So now here I'm calling this sequence. Execute biosec. Sec is not defined. First we need to define. Okay. Then call this biosec. So this is our sequence. Here you can see sec function. Now here itself call type function to check type of this attribute, this variable. So this is what bio.sec.sec. Okay, so you can read here detailed information. And also let's check what all functions are available with this bio.sec. So I'm calling directory function to check. So the which are existing with the string, and here with a sequence string or biosec string. Let's check the difference. So you'll get here different options like back transcribe, even reverse complement, which is not available in simple string. Reverse complement RNA. If we are working with RNA, we can use this. Then okay, So there are many other backgrounds and what else complement RNA. So you can see there are more advanced features or advanced methods are available with help of seq. Okay, so if you compare the both directories for biosec and sequence, you can see additional methods, you can see additional attributes which are provided by sec class. So definitely it helps to understand extra functionalities which biopython adds for this sequence manipulation. Okay, now uh, let's let me call or let me just I'll open this third session. From that, let me open which we already done, that is here I am pasting. So this is a sequence which we have created, but the change is what? Here I am using sec function. Remaining thing are same like transcribe use and complement, reverse complement, these all functions. So or before that, in the same cell I'm calling, just for your understanding, bio.sec. From that we imported sec class. Okay, so these all things we can do. Here I'm not using these all things because there are many more things from biopython we need to learn. So these are very basic. Here we have created one sequence, one string with help of sec class. That sequence, the sequence DNA sequence, we have transcribed. So we are getting here transcribed DNA. That is nothing but RNA sequence. Also, we translate the RNA into protein by using translate method. So we'll get this protein sequence in this variable. Okay, and after that, there's a complementary DNA and reverse complement. So let's execute this. Here it is. So you can verify this is the original sequence, conversion, that is central dogma, transcription, translation, protein, complementary, and reverse complement. Okay. So we are getting this message just because it's a protein sequence we are getting in triplet, right? But this is not a proper sequence, so a warning message will be there. So this is just for our understanding. Now counting base pair is also done. Let's check in this for same sequence. Suppose we want to count A. So first we need to call sequence and then count function. Okay. So let me check this variable name. Okay, or directly we can call sec or dnsec, any variable which you have. Let's suppose we are working with this sequence which is named by biosec, so you can call this variable and then count for a, count for t, atgc, anything. Okay, so counting and found, these all things we have covered. Is there any query in this part? Even we checked uppercase, lowercase. Okay, if you have any query or any point is not clear from that class which related to string manipulation, then please ask, we'll repeat that part.
Ma'am, I just have one doubt. Like for tomorrow's test, if I'm revising and if I get some doubt, would that be okay for you to, like, if I ask you something in the middle of like tomorrow, would that be okay for you?
Yeah, yeah, you can ask in the WhatsApp group is given, no? In, so
Yes,
you
ma'am.
can share your query, whatever query you are raising. Okay.
Okay. Also, I wanted to know, like, is this all or is there any kind of advanced coding stuff that we want to know that is related to Python and bioinformatics, and how can we find that, ma'am? Like, which websites do we have to go through to know other types of things that is
Okay.
advanced from this?
Okay, I'll share that links in, or I'll give the any if I have any PDF or something, I'll upload that in your given drive. Okay.
Okay, ma'am.
Yeah, and the tomorrow's test is like basic, from scratch. You need to explore like what is the data type and start from the initial. It's not like a hard level, but yeah, you should know the things which are very much required to understand any programming language. They are all basic questions.
Okay, ma'am.
Yeah, all the best. Don't worry about that. Okay, so now let's work with another class from bio module itself. So here I'm calling first bio. From bio, let's import entries. So here we can interact with online biological database. So like gene database or nucleotide protein database, PubMed, anything through entries. So how to work with this? So first, as it is an online database, you need to provide your email ID just for verification, or we can say just for tracking purpose. Okay, so here I'm providing, or let's first call this class. Now we are ready to work with this. So here call entries, here it is, and then provide emails. So you'll get these options. Email, here you need to provide just for identify yourself to NCBI, we need to provide our email. So here I am providing one email just for demo purpose, like this. Okay, now uh, if you know any ID, then you can provide the ID, otherwise you can provide the name of the thing, gene. So here I'll give one demo ID, like this is the ID. Let's provide in quotes. Then first thing, we need to create one handle, so that handle hold the information, so that information we can reuse multiple times. Okay, so we need to fetch information. So as I told here, in Entrace you can use efetch, esearch is also there for gene bank or PubMed. So first we'll fetch something, okay, by using efetch. So first let's create one handle. Here I'm giving handle as a name, variable name, call entries. Through entries, let's fetch. So call efetch. Here you'll get option. This is a efetch. You can call this as a function or method, and then provide the detail like which database you want to explore, or ID, or what type of here you'll get information like how you can write. Okay, so from bio, import entries, email ID, handle. So database is provided, nucleotide ID is given here. We have already provided ID. So for ID variable, ID parameter, we can call this gene ID variable. Retrieval type is gene, then retrieval mode is text. You can use text, JSON file, XML file. There are many types of file. And if you want to read the data line by line, so this is we already learned in file handling. Okay, read line, read lines, strip to new line character, switch to new line character. So these all things are there. So let's try one by one. In DB, let's provide gene, as we have provided gene name, gene ID. Then for ID parameter, this ID we can use, which is we already declared. So I'm using the variable name, or directly you can mention the 7175. Anything is okay. Then let's retrieval mode or anything. Let's take mode. So this gene ID for that gene which you want to fetch from NCBI, that all thing we have provided. Now we are fetching that gene data from NCBI in the XML file format. So let's see how to read that file format. This is just a demonstration, because this mail ID is not valid. Okay, here I'm providing, as we want to read from handle, so call that handle variable and use read function. If you want to read line readable, depending on that, you can call any function. Let's read. So call this. After reading this, you want to execute, right? So call print function and print gene data. That's much. So this will print the fetched gene data. Execute. Okay, so this is XML file format. You can see this web link is given, HTTP, and is all detailed, when and where, the date of that data it is submitted. So this is a big file. So this way we can interact with the online database through biopython. If you want to just fetch information about any database, so that also we can do. So let me copy this here. From bio, import entries. Also email, I'll keep the same thing, but as we want a database, so just change this all things. Here I'm writing database information. Now this time we'll fetch from protein information. So call entries, and from entries, as we are looking for information, so e-info function we can use. So from which database you want to fetch information, that database we need to provide. Recommendation is here, you can go through this how to write and all these things. Yeah, here it is written database list, pubmed, so we can use this. Okay, now let's read the information. So for that, the variable is db underscore info. So let's call entries. From entries, let's read this information. Okay, and then it will give you the detailed information. So there are many, it's like a key, a dictionary, it's like a key and value information. So let me call first db. We'll see what all attributes are there. So these many keys and their associated values are there, like you can go through this, or if you want specific only any particular key, like this, we can fetch information only. What is that information? So here we can call that inside square bracket quotes, is I think already there. Yeah, or we can mention this in round bracket. So this list we can provide. So only for information. What happened? Just wait. Let me check. It is showing full information. Okay, this is showing for DB info. There is no other information. You can see DB name, PubMed, and description. This is nothing but bibliographic data. Like for literature mining, we can use this. Literature mining means if you want to read or mine the information, any paper or relative studies, so we can go through this literature through PubMed. Or there's a meta analysis, like if you want to go with the review, search for the review or analysis for any existing research, then or simply if we want to perform automated workflow, like in bioinformatics pipelines, we need to set the automatically how to fetch the information from relevant publications, right? By using different criteria, we can write a keywords. So that all things we can fetch through this e-info from Entrace class from biopython. So this Entrace module basically is very important for literature and the data. For those who are already aware about this PhD literature review, they might aware like how it is useful to go through the information, author's information, publication date, study type. Okay, like supporting research material, you can fetch through this. Then okay, then if we are working with the sequence files like FASTA files or any kind of uh, files through biopython, if we want to read that file in this environment, so let's take example sequence only, FASTA sequence. So for that we need seq.io. Seq.io module is very widely used for reading and parsing the files like a FASTA files. So a FASTA file is aware, right? This format is aware, like first line starts with greater than sign, then id or information, and then accession id, the details, and then sequence. Second line onwards there is a sequence. Sequence are like broken into lines with the fixed length, typically 60 to 80 characters. Okay, this is not mandatory, but they are like a sequence for each line. So let's see how to fetch information from sequence. So first you need to import from bio. Let's import seq.io. Then only we can work. So if you have any file, you can use that file, or you can create here itself just by providing a new file and then write here demo.fasta like this, and you can create a FASTA file here itself. Otherwise, if you have any file, you can import that file by using this upload to session storage. Okay, or let me provide one file in chat box. You can create this file, few lines I'm providing. Okay, this much is enough. Okay, you can create a new file, FASTA or any demo.fasta, and then paste this data in that file and save. Okay. So from that sequence, let me open that sequence or any sequence. Like here, it is a protein sequence. So this is again greater than sign, ID, the name, and amino acid sequence. For I think this is DNA sequence. Yeah. So this is the DNA sequence, and this one file, this text file, actually contains a multiple sequences. Okay. So this file, if you want to save as a FASTA file, so let's copy these all things, and I'm also creating inside this as a demo.fasta. Fine. So this is one FASTA file we all have. So from this FASTA file, there are multiple sequences. And if we want to fetch only IDs, like from thousands of data, we want to just fetch IDs or just want to know the name, or if anybody is interested only in sequence. So how to segregate this data? First thing, how to read the data. For that, we need to use read.fasta function. And after reading, if we want to work with internal content of the file, like parsing of the file, so how to read? First thing, we need to specify path, which file we want to read. So for that, let me create here, or let me write first. This is just a path. Let's provide one variable to store that path. So here I'm writing, as this is a FASTA file, so let's write FASTA file, and then here we can provide the sequence path. So let's copy the path and provide address here. So this file we want to fetch. Now after that, we can pass the file by using secio. So for that, let's create one variable to parse and store the information called secio. From secio, we are going to call function parse. Here it is. Okay. So for parsing, first two parameters are required. First, name of the file and mode of the file. So which file we want to read? This file, because here we save the content. So first provide the file name and comma. The file is in FASTA file format. So mode of the file, type of the file. This is done. After that, we need to read that file line by line, because to fetch information, fetch ID, fetch description, fetch sequence, we need to parse. Parse means check line by line. So first, we need to create one iterator by using loop. We can go inside the file by, let's create one loop by using for keyword. For, let's create one iterator, or just a variable you can call. So here I'm using record. In where you want to iterate, in this records, because this records variable contain this file, as we are passing this file through secio. Okay, complete the first line, that is loop line, by using for with colon. Come to the next line. Now what we want to fetch? We want to fetch information for ID and description and sequence. So for that, let's call one by one. Print, either you can directly call this iterator record dot id, then record dot description, record dot sequence, or if you want to provide a proper label, then call print function. Print, this first information is for ID, so I'm providing label ID, and then fetch this record dot id. Okay, like this. So good representation. Then also for description, we can do same thing. This is for description, call record dot, there is a description. So let's select this. And also we can do the same thing for sequence. So print sequence and fetch parsed sequence by calling sec. Okay, so this way. And then when we want to execute that thing, so execution or reading, we can say, for reading, let's come out of the loop and then execute by calling sec.io dot read. So we can read the data which is stored in. This is not mandatory. Anyways, we are going to read by using this. So let's first execute this. Or this is another method, or let me give it for your information. Pasta file and then mode of the file. And then just simply execute the data. Okay. So you can see this is ID, description, and sequence. Okay. Like this. So first pass function is basically used to read FASTA file, and it returns the iterator of that sec record object. So we can represent the sequence from that file. And two parameter we use, that is FASTA underscore file. This is nothing but the file path which we store in that variable, and the FASTA. FASTA is nothing but the file format which we used. So record is a variable or iterator which is used to access each sequence or each record from that FASTA file. So there are two functions. One is sec.io.parse, which is used for multiple sequences. And if you just want to read a file having a single sequence, then we can use sec.read. Like here, I mentioned sec.read, sec.io.read. But here we use sec.parse because we want to handle multiple sequences from that file. Okay, so first we can use this for sequence annotation, when we want to read any large data set or genomic sequence. Even if anybody is working with a drug discovery and they want to analyze protein sequences just to identify drug targets, potential drug targets, or in genomic research, basically, if anybody is working with a variation or mutation, so that time need to parse or need to compare the multiple sequences, right? So in clinical diagnostic, in drug discovery, drug designing, we can use this parsing by using sec.io class in biopython. Then one more important application or use of biopython is alignment. So local alignment, global alignment. Are you aware about the alignment? Global. Global is basically used with Needleman and Wunsch algorithm, right? Means it's global means when we align two sequences end to end, means end to end means like having similar length. Okay, so that time specifically expected to be a global similarity. Wherein local alignment is what? Local means we are just looking for local region with help of Smith-Waterman algorithm. So local region between both two sequences. So when you want to study for any domains or motifs within uh, or just want to check the dissimilarities between sequences, so that time you can focus on local sequences. So here we'll work with pairwise sequences first. Pairwise means we need a pair, we need two sequences. So yeah, here itself we can create, you can create string and then convert into sec, otherwise directly you can create a sec sequence. So first we need that class. So again, from bio import pairwise 2. So this class is required. Execute this class and then perform pairwise alignment for two sequences. So we need two sequences. Let's create by using sec class. So here itself I'm providing sequence. This is suppose one sequence. Execute. Let me take this and create one more. So this is nothing but sequence 2. If you want to do some changes, you can just copy paste some stretch and paste it somewhere. So it is the same length, two sequences, sec 1 and sec 2. So now for alignment, let's create one variable to store the alignment result. So here, as we want to use pairwise class, so first call pairwise 2. From pairwise, we want to perform alignment, align, and then you can specify the method like global or local. So here I'm providing global. This is the way how you can write, and then provide for which sequences you want to perform. So these are two input sequence we have. Let's provide here sec 1 comma sec 2. Okay, and there is one more parameter you can add, like one alignment only. So that one alignment only basically we can use to specify optimal alignment. That optimal alignment as a result we are getting, so we can use this. Here you can read the detail parameter how to use, and sec a is what, sec b is what, score is what. So sec a and b we are going to use when we call the result, when we execute or when we iterate in that sequence to check the alignment. Then we are calling this sec a and sec b string, which shows the alignment between the sequences. So let's first uh, provide one parameter, one alignment. So we'll get optimal alignment. Let's keep this true. Okay, done. Then the actual part, that is we need to iterate in that sequence, in that one by one base, and then let's check the alignment because this is global alignment. So for, let's use one iterator alignment inside, or let's change the name here. Let me keep this alignment colon, and then execute the result. So first is nothing but iterator alignment dot sec a. Here you'll get sec a and sec b. Okay, so this way, or you can call here print, so it will execute the result like print. This is for first sequence. So let me write it like this: for sequence 1, and then fetch this way variable. Okay, like this. You can see your alignment from end to end. Okay, so in this pairwise to align dot global function, here global alignment of these two sequences, sequence 1 and sequence 2. So global x function, where simple scoring method is used, means it matches score is 1, mismatch and gaps are scored is 0. Are you aware about these terms, right? Match, mismatch, penalty. If not, then just read the alignment, different algorithms, and how to calculate the score, these all things. This is all theory behind this. Okay, so this way we are getting the result. And if you want to perform local alignment, this need to change with the local. So for that, or let me copy this, we perform local as well. So local access we can use. For local we can also execute score, like let me execute that. uh, Let's call string formatting. So score is nothing but base score. Okay, so or we can use this alignment dot score. We are fetching that value, so that's why in curly bracket. Okay, so this also we are getting. There's one more function called as a pairwise aligner. So for that, suppose, okay, let me copy this. So from same module, from bio import pairwise 2, we can also import align, and we can also import sec differently. Like here, let's call align for alignment, and sec, or from biosec we'll call sec, like this. This is for local alignment specifically. Okay, so when we want to perform pairwise aligner, we need to set the mode to the local. Means there are aligner mode we can define externally. Like here we directly call local access. So we need to call local or global, which mode we want to prefer explicitly. Like suppose let's create one variable. This is aligner. Mm, in aligner we can call align. To align, let's call pairwise aligner, this one, and then specify mode. So aligner dot mode, and then specify this here. Suppose we want to set for local, and the same thing, like you can fetch two sequences like this, or we can copy this here, these two same sequences, and then perform aligner dot align, because this is the align. Like here we use aligner dot mode, we can call here aligner dot align, then provide the sec1 and sec2 and execute the score. The same thing, you can try this pairwise aligner. So what exactly the difference between pairwise 2 and pairwise aligner? Basically both are simpler, but in pairwise 2, this is a old module, this one, uh, the function called as a align global, align local. So for quick alignment of local and global we can use this. So these parameters are more limited and less customizable. So we need to we are not getting we are not customize these all things. We are getting the default basic match, default mismatch or gap penalties, and in return we'll get that sequence A and B and start stop score everything. But in pairwise here, aligner, aligner is basically we can control the customization. Customization means we can set what exactly the penalty we want to keep, opening extension or mismatch match score, even offline gap cost if we want to specify. So these are all advanced customization option we can provide from user side. Okay, that is the thing only. But ease of use is align pairwise too. Even you can check the output format, it's different in both. Okay, now I think we have covered almost many modules or many classes from biopython. Any query till here?
Yeah.
Okay. So the similar way, any module you want to work with or any class, first you need to install biopython, then you can install the module, bio module, and from bio module, which class you want to work with, that class you can import. So you can work with that class. Even you'll get a detailed parameter description for that when you start writing. So take help of that or that document. So it is easy to write the code. Okay. So this is all about biopython. It's not easy to cover everything in one session for biopython, but yeah, like this module is for, like, from scratch we have started, so most of the part we have covered. Now you are, means, aware about how to explore python, how to learn or how to write code, so definitely you can do this. Okay, so successfully we have completed this python module with core concept and core application. So from understanding the basics like control structure, like how to work with libraries, how to work with a numerical data by using numpy, pandas, so you have built a solid foundation, I hope. And keep practicing, explore new projects, and also remember python is a gateway to endless possibilities in technology, in science. So all the best for your coding journey. And I'm here itself, any doubt, any query you have, you can ping. So we will be here to help. So
Thank you, ma'am.
yeah, so from Monday onwards we'll start our new module, R programming language. Are you aware, anyone of aware about this programming, R programming?
I know a little bit, ma'am.
Okay, so we'll start that also from scratch. Okay, then see you Monday.